My name is Li Tian. I'll introduce the paper submitted to iMeta. The title is Specificity Landscapes of 40 R2R3-MYBs Reveal How Paralogs Target Different Cis-Elements by Homodimeric Binding. In eukaryotes, paralogous transcription factors exhibit a specificity paradox, wherein paralogous TFs recognize highly similar DNA sequences yet regulate distinct physiological functions. This phenomenon has been reported in both animals and plants. The VIII R2R3-MYB transcription factors also face the specificity paradox. In this study, we employ high-throughput selects to analyze 40 R2R3-MYBs, revealing a homodimerization-based mechanism that enables eukaryotes to differentiate paralogous TFs. This work generates the largest plant selects dataset to date and identifies a highly specific AtMYB binding motif, termed the QAA-box. Our selects libraries included 38 VIII AtMYBs, 14 of which were not covered by previous datasets. Importantly, the selects libraries can offer more dimeric binding models. Moreover, the selects motifs cover a larger specificity space than the DAPSEC motif. We first examined the monomeric binding preferences of the VIII AtMYBs and clustered. Notably, the E-clade prefers to bind the AQUA sequence named QAA-box featuring a higher specificity than other AtMYBs. This difference is due to the mutations at positions 53 and 113, supported by the structural data analysis of M. polymorpha and S. Moellendorffi. Next, we further distinguish the AtMYBs by their homodimeric configurations. QAA-box AtMYBs 2, 57, and 79 show distinct homodimeric configurations in selects and DAP libraries. Notably, we observed that AtMYBs can change the DNA binding specificity when two monomers bind closely to each other. The site is different from the classic monomer, especially for AtMYB2, S or 1A. This modified specificity has not been reported and can change DNA shape. Moreover, we found that the non-DBD region affects the dimerization and DNA binding specificity of AtMYB2. Because AtMYB2 drastically changes its specificity, we next focused on R0, 1S, especially R1A, of AtMYB2 to explore. We examined the enrichments of R0, 1S in selects and DAP libraries. While most AtMYBs did not enrich R0, 1S, few AtMYBs did, and in selects library for AtMYB49, confirmed that this is a false positive. Thus, R0S and R1S are unique to AtMYB2. Meanwhile, we performed EMSA and observed that the dimeric binding was only for R1A and R8, not for MA, suggesting that R1A is a functional dimeric CRE for AtMYB2. Furthermore, DAPSEC of AtMYB2 have R1A peaks. Notably, the selects motifs of R0, 1S uniquely improve the prediction accuracy for AtMYB2 DAPSEC peaks. Altogether, the modified specificities allow AtMYB2 to recognize additional genomic sites. We further explored the roles of R0, 1S of AtMYB2 in transcriptional regulation. We found that R0, 1S are enriched around TSSS, and the reporter assays found that R0, 1S can greatly drive reporter gene expression. Similarly, the DAPSEC and RNA-seq data also supported this. Therefore, the R0, 1S CREs are functional to activate transcription. Consistent with it, the target gene of AtMYB2 expression increases after a BIA treatment, and the R1A consensus is perfect, which drove higher gene expression. In general, the GO analysis agreed with the reported functions of AtMYB2. Additionally, the AtMYB2 CREs are compatible with nucleosomes, indicating that AtMYB2 is a potential pioneer TF. Next, we used FASTkin scores to evaluate the conservation of AtMYB2 CREs. We found that the dimeric CREs co-localized with peaks of conservation scores. Mutating R1 as CAC positions narrowed the conservation peak, further supporting that the modified specificities of AtMYB2 are conserved in evolution. Finally, in this study, we analyzed 40 R2R3-MYB TFs by HT selects and identified a QAA-box AtMYBs. We revealed homodimerization has a novel mechanism enabling distinct paralogous TFs. Furthermore, the R1A of AtMYB2 is evolutionarily conserved, biochemically active, and transcriptionally functional.